Thanks everybody for joining us today. We're joined by Dr. Larry Hoffman, who received his PhD in physiology from UC Davis and is now an adjunct professor in the vestibular neuroscience lab at the head and neck surgery department at UCLA. And his principal interests are in neurosensory coding and primary afferent neurons, projecting from the semicircular canal cristae neutricle and how this coding can be modified, the dynamic specific projections, the brainstem and cerebellum, as well as the biophysics of Scarpa's ganglion neurons underlying the dynamic response diversity. We see a lot of patients with vestibular concerns, so we're excited to hear your presentation on the hype and peripheral vestibular hypofunction.
Well, again, I must start by thanking the House Institute Foundation and Steve for inviting me to present to you some of our work that we've been conducting over the years in peripheral vestibular hypofunction, and why we're so excited about it. And it's offering us a great deal of intellectual satisfaction, as well as hopefully we're moving the needle forward in the applications of our findings to helping you as otologists treat individuals with these disorders. So just in brief, I'd like to give you an idea of where we're headed here today. I'll start with an introduction and why we're motivated to do this work. I thought it was appropriate that I give you a brief primer on peripheral vestibular neurobiology. Obviously, we dig down into the weeds on this, and to help you understand the translational value of this, I think it would be appropriate to give you a brief introduction on the neurobiology of the labyrinth. I'll talk about some models of hypofunction and how we assessed it. These models include some spaceflight work that we've done, and also most recently in some ototoxin-induced hypofunction. And then hopefully I'll be able to convince you that this does have significant impact on a better understanding of hypofunction and how it's manifested in your patients. So we look at hypofunction as part of a continuum running from hypofunction through normal function. And in fact, we've actually worked on some models of enhanced function that we will not talk about today. But again, the peripheral labyrinth is a head-centric sensory system. It informs the brain something about what the head is doing to coordinate reflexes in gaze, and balance stabilization, etc. It's actually tuned to behavioral demands. So we found that there was differences across animal models running from the garden variety laboratory rodents through bats. We've done a little bit of work in some fruit bats. And so by virtue of looking at all this, we believe that this gives us insight into the factors that are driving the cellular features, which gives us a foundational understanding of how the bloody thing works. Why the hype? Well, I think it's a platform to enhance our understanding of the etiologies of the condition of hypofunction and how it might be manifested through a variety of pathways. It also could identify cellular targets for treatments. In other words, if you know what's gone wrong, perhaps there can be treatments that could be identified to actually rectify or remedy the situation. It also gives us some insight into what some of the functional targets for focused testing are, and that's based upon this foundational understanding of how the peripheral vestibular epithelia encode head movements. And then once we have an idea of what goes wrong, then perhaps we can develop more focused testing strategies. And of course, this gives us insight into the components of normal function. What are some of the strategies we use in pursuit of this hype? Well, again, we're looking at the cellular correlates of normal function. We use a variety of novel preparations elucidating the natural and adaptive capabilities of the peripheral vestibular epithelia. We principally look at the output of the epithelia, looking at the factors that contribute to these these trains of action potentials that are transmitted into the CNS by the primary afferent neurons, projecting from hair cells in the epithelia into the vestibular nuclei and brainstem. And along the way, we're finding out that this is unmasking some novel CNS processing strategies. And we won't be talking about that today, but we're very excited to be pursuing this work at this time. So this is probably at the level that you see a lot of your patients where they come in and perhaps complain of some imbalance or unsteadiness. And perhaps certainly here at UCLA, we have a longstanding history of evaluating eye movements. And of course, the eye movements are driven by, let's see if I can pull up a laser pointer here. Well, in any event, again, head movement stimuli evoke, sorry. Head movement stimuli evoke modulation in the afferent neurons, the discharge of afferent neurons projecting into the brainstem, and this illustrates the three neuron arc of the horizontal vestibulo-ocular reflex. And again, the principal task of this reflex is to stabilize visual gaze. Um, of course, this is driven by modulation of the activity of hair cells. And in this slide, this is an electron micrograph illustrating the different hair cell phenotypes that are found in all mammalian vestibular epithelia. We are a creative group, and so these phenotypes have the monikers of there's type 1 hair cells and type 2 hair cells. The type 1 hair cells are distinguished by something called an afferent calyx. So this is a specialization of the dendrites of these afferent neurons, and you can see it's illustrated here by the light shaded structures that surround these type 1 hair cells. Type 2s, of course, do not have a calyx. Down on the bottom here is illustrated the support cell nuclei. And a different rendition of these calyces is shown here where we immunolabeled at the afferent neurons with antibodies to calretinin, which is a calcium binding protein found in a certain region of the vestibular epithelia in all mammals. And then if we make that calyx a little translucent, you can see the hair cell inside. This is labeled by another calcium binding protein known as oncomodulin. And of course, type 2 hair cells are not endowed with calyces, but some of them do express oncomodulin as shown in this 3D reconstruction. So if we take a 10,000-foot view, this is a dorsal view of a mouse horizontal crista where all afferent neurons are labeled with antibodies to a microtubule protein known as beta-3 tubulin. And then again, as I mentioned a few minutes ago, a certain region, the central zone of the cristae in mammals, exhibit calyces that are positive for calretinin. And you can see that the expression of calretinin and calyces is restricted to this central region. We think of this as sort of like a fovea of the vestibular epithelia. It has particular physiologic properties that are unique to that region. We add in the oncomodulin staining, and you can see that those hair cells that are labeled with oncomodulin also label a particular area. Now, we understand why there may be the need for calcium binding protein presynaptically, but the role of calretinin postsynaptically is as yet undefined. We look at, again, one calyx in particular. Again, you can see that the calyx actually encapsulates the entire type 1 hair cell, and we can make that translucent where you can see the hair cell inside. So that's about all we'll talk about today on the morphology of the peripheral epithelia. So we'll transition to a brief discussion on how we assess the output, how we make direct assessments of labyrinthian output. And we do that in animal models, in rodent models, probing the superior vestibular nerve with high impedance microelectrodes where we can isolate the activity of individual afferents. we can the first thing that you notice when you drop an electrode into the nerve is the high degree of spontaneous activity. That is, activity of the afferent neurons that are present in the absence of any stimulus. That's exhibited here, where you can see that these action potentials in the top trace from the neuron from which that was recorded, the intervals in between action potentials is highly variable. And that's quantified by a parameter known as the coefficient of variation. And that's, in this case, this exhibits a CV of 0.6. And again, that has a mean interval of 15.2 milliseconds, which is about 60 to 65 spikes per second. The bottom trace illustrates the spontaneous discharge recording of a different afferent with a similar mean intervals, mean discharge rate, but the variability is much lower. And this is characterized by a CV of about 0.3. And that's just an illustration of the heterogeneity in spontaneous discharge. We call, just in terms to make the conversation a little bit smoother, we call the afferents that exhibit this highly irregular interspike intervals, irregular interspike intervals, or an irregular afferent. And the one on the bottom where the interspike intervals are much more regularly spaced, those are regular. That's a representation or an example of a regular afferent. We can also apply a variety of stimuli from discrete sinusoids to more naturalistic complex stimuli. And to illustrate the transduction cascade, my apologies. So when we apply these inputs, we record from the outputs. This is an example of two afferents. The per stimulus, or the record that was recorded during the application of a 3 hertz of a 3.2 hertz stimulus. You can see that on the top trace represents a recording of an afferent where the spontaneous discharge is recorded both before and after, and during the course of the stimulus, you see it modulates, and the vertical lines sort of highlight that the correlation of the peak response to the peak stimulus. So you can see that the response of the top afferent is quite noticeable. As it changes from spontaneous to an evoked response. However, in the more the afferent represented on the bottom, it's a little bit more difficult because there's the spontaneous discharge is much more variable. And this is quite a challenge for the brain. In other words, the brain, we know we can detect the response because we know what we put in. Of course, the brain doesn't necessarily know that, and so it has to decipher from this incoming spike train when the head is moved from afferents that have very regular spontaneous discharge, the transition to an evoked response. That seems to be a pretty easy task, but it's a much more challenging task for the more irregular afferents. Now, this is a representation of a similar pair of regular and irregular afferents from the same animal. And in this case, the regular afferent is shown in the blue circles, and the irregular is shown by the red dots. So this is what we call an input-output curve. The stimulus was a 1.6 hertz stimulus with a peak velocity of 15 degrees per second. And you can see that the variability throughout the response spike train. So this is the dots shown here represent the instantaneous discharge rate as the sinusoidal stimulus transitions from in a sinusoidal fashion. The variability in that discharge is very limited in the regular afferent, but it's much more highly variable for the regular afferent. So again, the red dots represents an afferent with a CV of 0.41, and so that would be characterized as an irregular afferent. But the CV of the blue dots of the blue afferent is much lower than that, and that's a much more regular afferent. The magnitude of the response is much different as well. The magnitude of the irregular afferent is higher, about twice the magnitude of the regular afferent. We've begun to use this parameter known as coherence. Coherence is defined as the as a linear representation of how well the response is of the stimulus is reflected in the response. So it's really a measure of response fidelity. As the coherence values get lower, you can interpret that as that there's other components of the response that are unrelated to the stimulus, and so therefore we would call that noise. So, for example, in this particular in this slide, the red afferent, you can see that the irregular afferent has much more per stimulus noise compared to the blue afferent. We can also characterize this in terms of the root mean square error, which is essentially similar to a calculation of standard deviation at a particular velocity. So at 15 degrees per second, the root mean square error for the regular afferent is shown as a little over 4, but the irregular afferent, the root mean square error is much greater. So the point here is just to illustrate that the more irregular afferents, while they have a much greater response magnitude, they're also endowed with much greater noise characteristics. And that's actually summarized in this plot where I've illustrated the root mean square error for the regular afferents and the irregular afferents of this sample of about 50 cells. And we tested the hypothesis that the root mean square error of the irregulars was similar to the root mean square error of the regulars. And the probability of that being true is very, very small. So we can conclude there that the root mean square error of the irregular afferents is greater than that of the regular afferents. And we'll come back to this a little bit later. So in summary of this brief introduction, um, uh, we can conclude that the calyx is a prominent component of the afferent dendrites. The discharge of afferent neurons represents a direct measure of stimulus coding. There's a broad heterogeneity among afferent neurons, and afferents exhibiting irregular spontaneous discharge interspike intervals tend to exhibit greater discharge variability relative to head kinematics. Furthermore, the stimulus response coherence is a measure of response fidelity to the head movement input, where high coherence reflects high response fidelity and a higher degree of certainty that a given action potential is actually a true representation of the head kinematic state. Whereas low coherence reflects lower response fidelity and greater uncertainty in the spike train. That is, there's increased noise in the response discharge. So what are the components that contribute to normal function? Well, certainly stimuli are transduced by the stereocilia. That depolarization of the hair cell gets modified by the conditioning of the receptor and generator potentials in hair cells and afferent dendrites. That leads to a modification of synapse function, both quantal and, as has been described over the last 10 or 15 years, non-quantal transmission between hair cell and the afferent dendrite. Now, again, the afferent dendrites is represented here as a calyx, but of course, there's other connections between the afferents and type 2 hair cells where there are no calyces. Again, the synapse function or transmission from hair cell to afferent can be achieved either through the release of neurotransmitter, and we call that quantal neurotransmission, but there's also direct communication, electrical communication between the hair cell and primarily calyces, and we call that non-quantal transmission. Because there is this non-quantal transmission, it's essentially similar to a capacitive coupling. The cleft now plays a very important role because that essentially is one of the components that establishes the capacitance between hair cell and the calyx. And of course, there's a reciprocal communication from the central nervous system back out to the epithelia, and those are known as the vestibular efferents. So I became interested in synapse function through my work with some space flight. So this actually dates this work because obviously there are no more shuttles that are being flown. So this was a flight project that I was engaged in about 15 years ago, but it was quite insightful. And the questions that we were asking is, does space flight representing a drastic attenuation in the ambient gravitational field, does that induce a form of sensory learning that we could detect through by quantifying the distribution of synapses? So this slide represents a cartoon, a series of cartoons illustrating how head movements impact the activity of or the displacement of stereocilia, hair cell stereocilia, both in the utricle primarily. You can see from the top two cartoons that head tilts backward and forward at Earth here on Earth. The otolithic membrane is endowed with a specific gravity greater than that of endolymph, and so therefore it's sensitive to the ambient gravitational field. So with a backward head tilt, you get slight displacements of stereocilia as the otolithic membrane tends to respond to the force of gravity, and there's very modest movements of the otolithic membrane and the stereocilia. Likewise, the opposite would occur with a forward head tilt. Of course, without head, you can also achieve displacements of the stereocilia in the conditions of without head tilts, and these are known as translational movements, where the head is accelerated and decelerated both in a nasal-occipital direction as well as in an intraoral direction. However, in the condition of microgravity, responses of the otolithic membrane to head tilt become negligible, if even non-existent, simply because the absence of the ambient gravitational field with head tilts would result in no displacements of the stereocilia. However, the response of the otolithic membrane and hair cell stereocilia to translational movements is still intact. So the best way to look at and quantify the distribution of synapses, of course, would be through electron microscopy. And this panel represents a stack that was collected through a technique known as conical TEM tomography, which gave us, at the time, it gave us 1 nanometer resolution. And you can appreciate that resolution as you go up and down through this image stack. You can see that the vesicles come in and out of the field of view. So this is pretty high resolution, and that was reconstructed in the cartoon on the right. Now, it turns out that we were hoping that we could actually glean information about the distribution of synapses. However, turns out that the aggregate or these cluster type synaptic ribbons that are seen here and are reconstructed in the red objects in the cartoon, you can see that those are cut off. So these clusters were much larger than we could achieve through the methods of conical EM tomography. That is, while we had great resolution, the depth through which we could sample was very, very limited. So we had to take a different approach. And to illustrate that approach, I'm showing you a top-down view of a mouse utricle shown here. And this is labeled with, again, antibodies to this calcium binding protein known as calretinin. So calretinin stains most of the type 2 hair cells throughout the epithelia. But again, as you saw earlier, it also stains the calyces in the specialized regions of the epithelia. In the utricle, this specialized region is known as the striola. And again, it has particular dynamic response properties that make it unique. Well, if there's a striola, that means that there's also regions that we can call the extrastriola, or regions outside of the striola. And so what we did is we used immunohistochemistry and stained for synaptic ribbons and a postsynaptic marker. And we used an imaging method known as confocal microscopy to sample the tissue and quantify the distribution of synaptic ribbons. So an example of a confocal stack is shown here. In white is shown the tubulin stained calyces. Obviously, this is a 3D reconstruction, and the red is immunohistochemistry for oncomodulin, and you can see that you have some type 1 hair cells that are encapsulated by these calyces, but there are other hair cells that are not encapsulated by calyces, and of course those are the type 2s. Our immunohistochemistry for the synaptic ribbons, though, showed a broad distribution of sizes, both within hair cells and associated with type 2 hair cells, and those are shown as the distribution of green dots. So in practice, what we did is we counted a synapse as those where the synaptic staining for the synaptic ribbon, which is staining positive for this transcription factor known as C-terminal binding protein 2, was found to be apposed or closely associated with a postsynaptic marker known as, and the one that we used at the time was a scaffold protein known as SHANK1A. And of course, the blue is the stains the nuclei. So this is done from a cryosection through the utricle. An optical section done with confocal microscopy shows something similar, where SHANK1A in this micrograph is shown in green, the CDBP2 is shown in red, and wherever we saw a coupling of the two, we counted that as a synapse. To give you an idea of how we sampled through the epithelium, we sampled 5 regions, 3 through the striola, 2 in the medial extrastriola, and this region outside the striola. And we also imaged the horizontal crista as a labyrinthine, as a representative of labyrinthine epithelia that would be insensitive to changes in ambient gravity. So this is essentially an intrinsic control to control for other factors associated with spaceflight, such as vibration, cosmic radiation, et cetera. Anything else that might impact the epithelium in general would be reflected in the horizontal crista, but it's only those that are those samples through the utricle where would exhibit some sensitivity to the changes in the ambient gravitational field. So what we found was that in the horizontal crista, there was no change in the distribution of synapses, quantified as the synapse density per hair cell. In the striola, there was also no change in the microgravity condition compared to controls. And these are Earth gravity controls that were maintained at Kennedy Space Center, and they were maintained in the same vivarium housing as was aboard the shuttle. However, in the medial extrastriola, we found a dramatic decrease in the synapse density. That decrease amounted to about 30%. So it sort of illustrates that when you remove the ambient gravity, or one important component of the forces that are responsible for activity in utricular hair cells, if you eliminate that, synapse density diminishes. So we consider that a form of synaptic plasticity. Interestingly, there was a study produced by a European group whereby they assessed utricular function through ocular counterrolling measures. So they place a chair at the end of a centrifuge, and then they superimpose a rotation at the end of that gravitational field, and that gives them a measure of utricular function. And they can do that with either clockwise or counterclockwise rotation. And the fact of the matter is that the ocular counterrolling in cosmonauts, after spending 6 months aboard Mir, the Russian space station, and this data was collected over a period of decades, but the finding was that there was essentially a 30% decrease in utricular function upon return to Earth. So this was before flight. This is return plus 2 to 3 days. And then by the time the cosmonauts had returned for 9 days, essentially ocular counterrolling or utricular function had returned to normal levels. So again, we have no idea whether this is represents a form of is reflected as a synaptic plasticity in these cosmonauts. However, it does illustrate that spaceflight is associated with a vestibular hypofunction, potentially as a form of synaptic plasticity. So it appears as though that the synaptic plasticity conforms to a learning rule that we characterize as use it or lose it. It suggests that hair cell synaptic plasticity reflects alterations in the sensory demand, both static and dynamic. The fundamental mechanisms by which this plasticity is achieved is completely unknown. We don't know whether it requires input from the CNS through efferent neurons, or if it is a factor that is intrinsic to the sensory epithelia. But it does raise the question as to whether synapse densities can be enhanced. And this will be important as we move through the rest of my presentation this morning. And therefore, we're addressing questions of hypofunction and whether that reflects a form of synaptic plasticity. So in terms of identifying the components of hypofunction, we're focusing on synaptopathies. And many of you have probably heard of the synaptopathies that have been found in the cochlea in response to acoustic trauma or even aminoglycoside ototoxicity. So in terms of the models that we use to study these vestibular synaptopathies and associated hypofunction, I've illustrated some work that we've done in spaceflight, and as a little bit of a teaser, I was also involved in another flight project over the last couple of years, which will provide further insight onto these mechanisms. I'd also like to present to you a little bit of data from not my lab, but another lab on aging. This came from the laboratory of Gabe Korfas at the University of Michigan, whereby he looked at aged mice and he looked at the distribution of synapses. And the bottom line is that 24-month-old mice did exhibit a decrease in the distribution of synapse densities represented by immunostaining by this, again, the same transcription factor that we use, the CDBP2. And that actually had a functional correlate where they use a vestibular evoke potential, which is similar to an ABR. It's evoked by a stimulus transient, and it relies upon the synchronous activation of a population of hair cells and afferent neurons. And they found that the threshold of this vestibular evoke potential was elevated in the same animals where they found a decrease in the synapse density. So what other models are available for us to study? Well, we began using an ototoxin-induced hypofunction about 10 years ago. And just to remind you of this measure of coherence, this panel illustrates the heterogeneity that is found naturally in these coherence measures in afferent neurons projecting from the horizontal semicircular canal. The stimulus here is, again, the 1.6 hertz, 15 degrees per second stimulus. And you can see that in these response waveforms, the top representation has a much higher coherence than that one shown in the middle trace. And you can see that there's various anomalies in this response waveform. Little dips and valleys that occur throughout. And these would be essentially not represented in the stimulus, so it would compromise this coherence measure, essentially representing a little bit of an introduction of noise into the response spike train. So how might ototoxicity induced hypofunction be achieved through the administration of aminoglycoside antibiotics? So this is a representation from this publication, and this reflects a head impulse test of a subject that had received intratympanic gentamicin for the treatment of intractable vertigo. The solid black trace represents the head movement, and the gray represents the eye movement. And what you do is you compare the peak head velocity to the peak eye velocity, and you can see in this case that the eye velocity only achieves about half of the peak head velocity. So we would call that the gain, or the response magnitude of that would be about half what it should be. Again, normal VOR, you would find that the eye velocity matches head velocity, but in this case, it's reduced to about 50%. And of course, these traces represent the catch-up saccades that the eye makes during this in cases of vestibular hypofunction. So what we did is, as you probably are aware, for decades, people were introducing gentamicin systemically. In other words, giving intraperitoneal injections and then hoping that some of that get into the ear. When I started this, I thought that was we could do better, and we could introduce the gentamicin much more locally, similar to what had been done with intratympanic, but even much more local than that. And to do that, we took advantage of a morphologic advantage of the chinchilla middle ear, and we were actually able to put a cannula down into the superior canal, not so that it either blocked or compromised canal function, but but it just actually allowed us to deliver gentamicin directly to the perilymph. And that's illustrated in this cartoon. And you can see that when we introduce the gentamicin through a syringe pump directly into the perilymph, it distributes throughout the perilymph. We found that the sweet spot for us at the time was just delivering 1 microgram of gentamicin, which conforms to about 130 micromolar final concentration, considering an estimate of the perilymph volume in the chinchilla. And we waited a variety of durations for this material to distribute and have its impact on vestibular function. So again, this panel will represent a representation of a healthy superior canal afferent and a horizontal canal afferent using this measure of coherence. And you can see that the coherence measure is actually quite sensitive, despite the fact that the magnitude of this response is very, very low. The analysis provides is very robust to even low magnitudes. It's based upon the spectral distribution of components in the response spike train. And you can see the coherence of the superior canal is actually very high, despite the fact that the magnitude of the response is very small. The horizontal canal afferent, this particular afferent had a greater response magnitude, and of course it resulted in a coherence measure was much larger. So in one animal, what I did was I recorded 10 consecutive afferents in response to that same stimulus. That stimulus was about half hertz, 0.4 hertz, and 30 degrees per second peak velocity. And we measured coherence during the per stimulus discharge of these afferents. Now, in ordinary conditions, in untreated conditions, the portion of the vestibular nerve from which I record, you will always find canal afferents, and you will always find responses. This is 10 consecutive afferents in which you can see that there is no response in any of these afferents. Coherence measures is quite low. And so, but however, this does represent that the neurons were firing. In other words, this represents a measure of spontaneous discharge. So in this case, the 1 microgram intraperilymphatic gentamicin induced a sensory paralysis. However, the spontaneous discharge was preserved, and that was quite significant. What were the morphologic findings in these animals? So again, we're looking at a top-down view of a chinchilla crista stained with calretinin, as you saw before for the mouse crista, and it's in this black and white, in this grayscale micrograph. You can see that this would represent the central zone of this chinchilla crista. This is an immunostained high-power view of that same crista, where this is an untreated specimen. Again, the beta-3 tubulin is the antibody that labels all calyces, and calretinin will label only those calyces that are shown in the central zone, that are found in the central zone. And you could get an idea of the degree to which these calyces are packed in. And of course, the calyces represent whenever you see a calyx, there's a type 1 hair cell there. However, with 1 microgram gentamicin, we found that all the calyces were lost. Every one of those calyces is gone. However, as you saw before in the previous panel, spontaneous discharge is preserved, indicating that the hair cells are still there. They're still releasing neurotransmitter. However, all the calyces were lost in these 1 microgram preparations. That's shown again in another view in this panel. In this panel, we're staining the hair cells with myosin 7A, which is a hair cell specific myosin. And again, we've co-stained with these antibodies to calretinin to illustrate the calyces in the central zone. So this is from a contralateral ear in a specimen in which we delivered 1 microgram gentamicin to one ear, and we sampled the other ear just for morphologic studies. And this is what we found. Again, the calyces have completely retracted. All the calyces are gone. The parent axon is still there, so it didn't kill the afferent, but essentially it rendered it basically eliminated or caused the calyx to retract from the type 1 hair cell. Again, hair cells are still there. Now, we do see a little bit of hair cell loss, about 20 to 30% hair cell loss, but the majority of hair cells are still there, and certainly many of them were type 1 prior to the introduction of the gentamicin. So, to summarize, we found that refined dosing optimizes our capabilities to create defined lesions. The hypofunction results in this case was represented as a dynamic sensory paralysis, whereby spontaneous discharge was preserved, but quantal and therefore quantal synapse function was preserved, but had a profound postsynaptic impact on the calyces. And so I'm not suggesting that all the impact was on the calyx, because again, we suspect that the gentamicin did have an impact on the hair cells. It didn't kill the hair cell, but it certainly rendered a dynamic response mechanism. It paralyzed that. But the bottom line is that this provided a platform for further investigation, and that's what we're in the process of doing now. So in this panel represents, again, 4 afferents showing the coherence measures on the right, and the response discharge at every symbol represents the timing of a spike, and the fits are the instantaneous discharge rate shown. And you can see that the distribution of coherence values to this 1.6 hertz stimulus. However, when we introduce either 0.5 or 0.75, we found that for the most part, coherence values were compromised. Again, it didn't eliminate, it didn't paralyze. These lower doses of gentamicin didn't paralyze the epithelium, but it introduced some measure of hypofunction. And again, we know that these are functional afferents. You can tell quite easily during the course of an experiment, and the analysis suggests that even with a coherence value of 0.61, that there still is a response there. It's just that it appears as though there's an introduction of a great deal of noise into that response spike train, and the same can be said for the 0.75. So when you actually look at the magnitude of the response of those afferents, we found that there was no difference between the treated and the untreated afferents. And we used a test of relative entropy to do the statistics there. And we basically found that you couldn't, that the likelihood of getting these response magnitudes from these untreated specimens, you could have drawn it from the same population as the untreated. And so therefore, there's really no difference in those two, just the distribution of sensitivity measures in those two populations. However, we found that coherence was drastically compromised, and whereby using the same statistical test, we found that the coherence measures in these gentamicin-treated specimens as a population were drastically reduced. We are currently in the process of conducting the analyses of the distribution of synaptic ribbons in these specimens. And so more to come later on that. So what I'd like to end on is a recent finding coming from Rick Lewis's lab at Harvard. And what they did is they looked at NF2 patients with NF2-related schwannomatosis, as well as patients exhibiting spontaneous or sporadic vestibular schwannoma. And they compared the VOR gains amongst their normal cohort of subjects, the sporadic schwannoma cohort, as well as the NF2 cohort. And they measured VOR both with stimulus sinusoids of approximately 1 hertz, as well as the vestibular head impulse test. What they found is that the gain magnitude of the NF2 was essentially the same as their normal subjects. So there was no difference between the gains in either, again, sinusoidal stimuli or the head impulse test between the NF2 and the normal subjects. And this they use gain as a proxy for response accuracy. And so they concluded that the accuracy of the NF2 patients was intact. However, there's a much greater variability in the NF2 patients, much greater variability. And they characterize that as a decrease in precision. And through a lot of very exquisite work done in preparation for this study, done by one of the co-authors by the name of Faisal Karmali. Faisal has done a lot of work to show that to allow him to hypothesize or postulate that the increased variability, decreased precision, is due to an increase in the peripheral noise. So an increase in the peripheral noise. Interestingly, they had two patients that were undergoing treatment with bevacizumab, which, as you know, is an antagonist, an antibody antagonist to vascular endothelial growth factor. And again, what they found was that the standard deviation, so the pre-treatment VOR measures is shown as the open symbols. The post-treatment measures are shown in the filled symbols. And the standard deviation, so this is reflects the standard deviation of the eye velocity. And again, the standard deviation in their cohort of NF2 patients was lower than that of the vestibular schwannoma, or I'm sorry, the standard deviation was higher than that of the normal patients. So again, the precision was decreased. However, in both of the bevacizumab treated patients, the standard deviation of the eye velocity of the VOR measures actually decreased. So they saw that as a positive sign. So again, the treatment results in a decreased variability or an increase in precision. So in summary, what I've illustrated to you is that in our hands, in our measures of directly taken from the afferent neurons, that the sensitivity of the gentamicin cohort is approximately the same as the untreated cohort. However, the coherence measures are much lower. And again, the coherence is a measure of signal-to-noise ratio, where noise is a component of the spike train that is unrelated to the stimulus. Okay, so it's an introduction of noise. Apparently, gentamicin is having this impact to increase the level of noise in the spike train. A decrease in coherence essentially is interpreted as a decrease in the probability of a spike occurring with any given application of head kinematics. This can be interpreted as a greater uncertainty of labyrinthine output relative to the head movements. And we don't know that in our animal models, we don't know yet whether this is manifested in any head movement related behaviors, such as a head stabilization capability or the VOR. So again, further, the peripheral vestibular hypofunction is associated with synaptopathies of multiple etiologies, could be manifested through a decreased ribbon densities, and this suggests to us that this represents something akin to hidden auditory loss, or this may reflect a hidden vestibular loss. And this peripheral vestibular hypofunction is also manifested as diminished afferent discharge coherence to any given stimulus, whereby alterations in spontaneous or evoked discharge could be responsible for this. And again, diminished coherence or increased variability, decreased precision could be as a result of the decrease in magnitude or an increase in the noise. But what I've shown you is that the magnitude of the response, the accuracy of the responses has not changed. So it appears as though the culprit is an increase in the noise. So again, I'd like to acknowledge those that have contributed significantly to the work. David and Johnny were postdocs in the lab over the years. Amy Tan and Terry Prinz were graduate students. Kevin was a research associate who recently left the lab to pursue a PhD in neuroscience at Pitt. Ahmed is currently a postdoc in the laboratory, and he's responsible for much of the noise analysis that I've shown you. And Shireen also recently joined the lab as a research associate. I'd like to acknowledge my longtime UCLA collaborators, Yvonne Lopez and Felix Schweitzer, and my collaborator of more than 30 years at the University of Otago, Michael Pollan. And Mike King has been very instrumental in helpful discussions. And these are the individuals responsible for allowing us to do this work. So I appreciate your attention and thank you very much.